Hello everyone, I am Mushtaq Ali, presenting an acoustofluidic-based level-free and selective separation based on traveling surface acoustic wave. So here is a brief summary in the form of abstract. Here I have presented a schematic of my device in which I have applied the traveling surface acoustic wave produced by scientific RTT and separated droplets of different kind. So when these droplets experience a traveling surface acoustic wave, the acoustic radiation force are produced at the droplet interface. And when we have something inside the droplet, in addition to the droplet, the acoustic radiation force also produced on the encapsulated sample. So depending on the size of the encapsulated sample, the lateral migration distance of these droplet will be different, and depending on the property of the sample encapsulated droplet, the lateral migration distance of these substance we expect it will be different. Here droplet microfluidic has been introduced with their few advantages and applications. Existing droplet separation techniques offered by optophoresis, electrophoresis, magnetophoresis, and acoustophoresis either need labeling or detection of in-droplet sample for the droplet separation. In addition to the labeling and detection, the biological sample existed in vast sizes, and conventional droplet separation techniques are typically limited to droplet separation containing relatively large biological samples, and our approach overcome this limitation, enabling the manipulation of droplets containing much smaller entities, for example bacteria and platelets. So our droplet separation mechanism is solely rely on the sample size in relation to the droplet size. So when this sample size is much smaller than one third of the diameter of the droplet, the acoustic radiation force acting on the overall droplet is not significant enough to produce additional lateral migration distance. So this work has been published by our group previously. So in our work, we have modeled the acoustic radiation force acting on a droplet and acoustic radiation force acting on a particle differently. So since the droplet size is much larger than the acoustic wavelength, so we have used the ray acoustic approach to model the acoustic radiation force acting on a droplet. But the acoustic radiation force acting on the elastic particle is modeled using the the elastic sphere theory. We systematically examine the migration of droplet containing microparticle of different sizes under various acoustic frequencies. For example, when you use 38.8 MHz frequency, which is the effective frequency of 31.4 micrometer particles, so these PMMA particles, particle-laden droplets, produce greater lateral migration distance compared to PMMA 19.2 micrometer laden particles. Compared to these results, when we use the effective frequency of 19.2, which is 44.5 MHz, so these particle-laden droplets exhibit greater lateral migration distance, even though the sizes of these particles are smaller as compared to 31.4. So this is because these elastic particles resonate at certain frequencies depending on their size and material property. We also observe the material specific behavior. For instance, at effective frequency of 38.8 MHz, the PMMA 31.4 micrometer lead-in droplets experience greater lateral migration distance as compared to PS 31 micrometer lead-in droplets. Compared to this scenario, when we use the effective frequency of PS 31 micrometer lead-in droplets, so these at the effective frequency of 31.5 megahertz, so these droplets experience greater lateral migration distance as compared to PMMA 31.4 micrometer. So this is because the elastic particle resonate at certain frequency depending on the size and material property of these particles. Finally, we demonstrated the separation of droplet containing a small biological sample such as E. coli and platelets by encapsulating co-encapsulated polymer microparticle as a physical marker. So this strategy avoids the direct manipulation of delicate biological sample while maintaining high purity and efficiency. In conclusion, our label-free, detection-free acoustofluidic method enables simultaneous, selective, and multi-type separation of cell-encapsulated droplet, in, expanding the possibility of droplet-based microfluidic in both biological and medical applications. So if you have any question, you can email me or contact me at this number. Thank you so much.